A creature the size of a grain of rice drifts through open water. It has no eyes. It has no brain in any functional sense. It has a set of chemical receptors and a body built for one purpose. It finds a fish. It attaches to the gill. It begins to feed. Over the next several weeks, it grows, molds, and changes form. It is no longer a free-swimming larva. It is a resident. It taps into the fish's blood supply and draws nutrients continuously. The fish does not die. The fish swims, feeds, and behaves mostly as it did before. Mostly. There is one difference. The fish now spends more time near the surface. It moves into shallower water, into better lit zones where it is more visible from above. A bird spots it. The bird dives. The fish is eaten. Inside the bird's gut, the parasite releases its eggs. The eggs pass into the water. The cycle begins again. The fish did not choose to swim toward the surface. Something the size of a grain of rice made that decision for it. This is not an anomaly. This is the dominant strategy. Estimates vary, but the most commonly cited figure in parasitology is that roughly 40 to 50% of all known animal species are parasitic. Not marine species, all animal species. In the ocean, the proportion is at least as high, and possibly higher, because marine food webs are longer and more interconnected than terrestrial ones, which means more opportunities for parasites to insert themselves at every link. A food web diagram that includes only predators and prey is not a complete picture. It is a picture with half the connections missing. The parasites are the other half. They link species that would otherwise never interact. A worm inside a fish connects that fish to a bird. A larval stage inside a snail connects the snail to a crab. The web is not a chain, it is a tangle. And the parasites are the threads that hold the tangle together. Most ocean parasites are invisible to the camera. They live inside their hosts. They are small. They are not photogenic. A predator has teeth, speed, drama. A parasite has patience, chemistry, and a body plan optimized for attachment and reproduction. There is no documentary equivalent of a whale hunt for a worm that sits inside a fish's liver for 3 years, slowly redirecting where that fish swims, what it eats, and how visible it makes itself. The strategy is silent. The results are not. There is one parasite that is impossible to ignore once described. It targets a specific group of fish. The larval form enters through the gills. It crawls along the gill chamber toward the mouth. It attaches to the base of the tongue. It feeds on the blood supply that feeds the tongue. The tongue, deprived of circulation, atrophies. It withers. It disappears. The parasite, now roughly the size and shape of the missing tongue, takes its place. It attaches its legs to the stub where the tongue was and remains there for the rest of its life. When the fish eats, the parasite intercepts a portion of the food. The fish survives. It feeds. It functions. It simply has a crustacean where its tongue used to be. The parasite is the only known organism that functionally replaces a host organ. The fish does not appear to be in distress. It feeds normally or close to it. The parasite feeds when the fish feeds. The arrangement is stable. It is also, by any standard, one of the most precisely engineered acts of body occupation in the animal kingdom. But the tongue replacement is mechanical. Other parasites operate at a deeper level. They do not replace organs. They alter behavior. Trematodes, a group of flatworm parasites, have life cycles that require multiple hosts. A typical cycle moves through a snail, a fish, and a bird, in that order. The parasite needs each host for a different stage of its development. The problem is transit. How does an organism with no eyes, no brain, and no ability to swim get from a snail to a fish to a bird? The answer is manipulation. Inside the fish, the trematode larva insists in the brain or in the tissue behind the eye. The fish begins to behave differently. It flashes its belly near the surface. It jerks. It moves erratically in shallow water. To a bird, the fish looks injured. Easy prey. The bird eats the fish. The trematode reaches its final host. The behavioral change is not accidental. It is targeted. The parasite produces compounds that interact with the host's nervous system. The fish does not know that its behavior has been hijacked. The bird does not know it is being fed by a worm the size of a pinhead. The worm does not know anything. It is running chemistry. The result looks like intention. It is not. It is selection. Every generation of trematode that was slightly better at making fish behave conspicuously left more offspring inside more birds. Over millions of years, the chemistry became precise. There is a parasite that goes further. It does not merely change behavior, it rewrites the host's body. It targets crabs. The larval form, barely visible to the eye, drifts until it encounters a crab. It attaches to the outside of the shell, finds a joint where the armor is thin, and injects a soft mass of cells through the gap. Inside, it grows a network of root-like filaments that extends through the crab's entire body. The filaments penetrate the nervous system, the reproductive organs, the digestive gland. They wrap around the neural ganglia. They infiltrate tissue the way a fungus infiltrates wood, slowly and completely. The crab stops molting. It stops growing. Its metabolism redirects. If the crab is male, its body begins to change. The abdomen widens. The reproductive structures reconfigure to resemble those of a female. Hormones shift. The crab begins to behave like a female carrying eggs. But the eggs are not eggs. They are the parasite's larvae, held in a sac on the crab's underside. The crab tends them. It fans water over them to keep them oxygenated. It protects them from predators with the same intensity a female crab protects its own young. The crab has been chemically and structurally converted from a host into a nursery. It will never reproduce again. It will spend the rest of its life, which the parasite regulates to be as long as possible, raising the offspring of something that entered through a joint in its shell. The question that parasitologists keep circling back to is biomass. In a given marine ecosystem, how much of the total living tissue is parasitic? The studies that have attempted to measure this are few, because counting parasites requires dissecting hosts, which is labor-intensive and difficult to fund. But the results are consistent and unsettling. In 2008, a team led by Armand Couris at the University of California published a study on the Carpinteria salt marsh, a well-mapped estuary in Southern California. They counted every parasite in every host species across the entire system. The total parasitic biomass exceeded the biomass of all the birds in the marsh. It exceeded the biomass of all the fish. It was comparable to the combined biomass of the top 3 predator groups. Worms smaller than a fingernail, living inside snails and fish, collectively outweighed the herons that hunted those fish. The finding was not a quirk of one marsh. Similar results appeared in studies from New Zealand, Mexico, and the Gulf of California. Parasites are not a minor fraction of the living tissue in a marine ecosystem. They are a substantial fraction. In some systems, the dominant fraction. The deep ocean adds a layer of uncertainty. Parasites in the deep sea are poorly studied because studying them requires collecting hosts, bringing them to the surface, and dissecting them before decomposition destroys the parasites inside. Many deep-sea parasites have never been described. They exist in specimens sitting in museum jars, unexamined. The life cycle of a deep-sea trematode, if such a thing exists at 3,000 meters, would involve hosts that have never been observed alive in a food web that has never been fully mapped. The 5% statistic from the last video applies here in its sharpest form. We have not explored the parasites of an ocean we have barely sampled. The invisible fraction of an invisible fraction. If parasites were removed from a marine food web, the web would not simplify. It would collapse. The links between species that parasites maintain, the behavioral modifications, the population controls, the energy transfers between trophic levels, all of it would vanish. Parasites regulate host populations. They prevent any single species from dominating. They redirect energy flows in ways that keep the web diverse. Remove predators from an ecosystem and the prey population explodes, then crashes. Remove parasites and something subtler happens. The connections between species dissolve. The system does not overpopulate, it disintegrates. Parasites are not passengers, they are structural. There is a reason this goes unfilmed and mostly unmentioned. Parasites trigger disgust. They are small, they live inside other animals, and they do things that make people uncomfortable. A predator kills and eats. There is a narrative to that. The chase, the strike, the meal. A parasite enters, alters, occupies, and redirects. There is no chase. There is no visible drama. The host looks normal from the outside. The parasite is invisible. The story only emerges through dissection and chemistry, through life cycle diagrams that require 4 hosts and 3 habitats, through papers published in journals that most people will never open. The asymmetry extends to funding. Marine biology research budgets are weighted toward charismatic megafauna: whales, sharks, sea turtles, coral reefs. Parasitology receives a fraction. Most marine parasite research is funded through agricultural or medical pathways, because parasites that infect farmed fish or human shellfish have economic consequences. Parasites that live inside a deep-sea snail and alter the behavior of a fish that feeds a seabird do not have an economic lobby. They have a life cycle that is more complex than most engineering systems and a body smaller than a comma on this page. They are the most successful survival strategy in the animal kingdom, and they are almost entirely hidden. 600 million years of sponges surviving by doing nothing. Tube worms surviving by partnering with bacteria. Vents surviving by running on chemistry instead of light. And parasites surviving by rewriting the biology of other organisms from the inside. The ocean keeps producing answers to the question of survival that look nothing like what we expected. We expected teeth, speed, size, intelligence. The ocean produced patience, chemistry, attachment, and invisible control. Every food web diagram ever drawn in an ocean textbook is incomplete. It shows what eats what. It does not show what is being controlled by what. The lines between species on the diagram are drawn by predation. The lines that are missing are drawn by parasites. If they were added, the diagram would not be a web. It would be a circuit board. Every node connected to every other node through organisms too small to see and too complex to summarize. Somewhere in the water right now, a fish is swimming toward the surface. It does not know why. It is being steered by something it cannot see, something that entered through its gill weeks ago and settled behind its eye. The fish looks healthy. It is swimming into the light. A bird is circling overhead. The parasite does not know any of this. It does not need to.